Hello, welcome to Medical Lab Science Quick Review. We'll introduce you to the lymphoid family of cells with lymphopoesis, individual lymph cell types, and their specific, interesting, and important roles in our immune system. An informative Q&A session will follow this presentation past the 8-minute mark of this video. Hematopoiesis transitions early in life from the yolk sac and embryonic mesoderm to fetal liver and spleen, then to the bone marrow, where B cells mature and produce immunoglobulins. Let's take a brief look at lymphopoiesis. You may have seen this classic version showing lymphopoietic lineage. On the other side of hematopoiesis, we see myelopoiesis. Lymphocyte precursors migrate to lymphoid tissues and lymph nodes, thymus, spleen. Immature lymphs, such as the common lymphoid progenitor cells, migrate to the thymus to develop and differentiate into mature T lymphocytes, which includes three main types. The first type is the cytotoxic lymphs, CTL, also known as a killer T cell, also known as CD8 positive T cell, a potent weapon that recognizes and destroys virus and some bacteria infected cells and some cancer cells as part of a cell mediated adaptive immunity. Because of T cell receptors, they recognize antigens bound to the major histocompatibility complex class 1 molecules. The second type is the helper T cell, the one that directs other immune cells by way of cytokines like interleukins and interferon gamma. The third type of T cell is the regulatory T cell, TREG, which keep the immune system from overreacting into a hypersensitivity, which you may have learned in our other video. In flow cytometry, mature T cells are CD3 positive, but as you can see, the CD4 marker is also found in the T helpers and TREGs, and the CD8 marker is found on the CTLs. The hematopoietic stem cells inside the bone marrow are the primitive master cells that are capable of rebuilding almost any damaged tissue and differentiate into intermediate cells called progenitor cells that will later mature into two main branches. Natural killer sounds like a horror or drama movie, but natural killer cells are so named because of their innate ability to kill target cells without prior exposure to them. NK cells do not require the thymus for development. Instead, they mature in the bone marrow from common lymphoid progenitor cells. Like neutrophils, NK cells are on call, residing in the blood, spleen, and liver, some in the tissues, patrolling and ready for immune defense. NK cells make contact with abnormal antibody coated cells, perforating them with perforin, then releasing granules with granzymes that lyse virally infected cells or tumor cells, causing apoptosis. Likewise, NK cells attack and kill bacteria, parasites, and fungi as a first line of defense. NK cells also sound the alarm, secreting cytokine signals such as interferon gamma that can activate and summon macrophages to attack. NK cells are identified in flow cytometry as CD16 and CD56 positive, the former as a surface marker for antibody receptors. NK cells are able to engage abnormal antibody coated cells that have abnormal protein markers that make them a target. As we can see, cytotoxic T cells are not natural killer cells. They are also known as cytotoxic T lymphs or CTLs or killer T cells. Cytotoxic T cells have T cell receptors and two potent weapons, perforin and granzyme. In flow cytometry, CTLs are identified as CD8 positive. Cytotoxic T cells are more antigen specific than natural killer cells because CTLs are driven by TCR or T cell receptors that provide a sort of two key system for immune safety and regulation. So, activation is double checked and activated at the appropriate time and place. Helper T cells do not kill or phagocytize infected cells or microorganisms, yet they are arguably the most important cells of the adaptive immune response because of the important role of orchestrating the immune response with cytokine secretion, driving other immune cells to activate and mobilize for attack. B cells become either memory B cells or plasma B cells, which reside in the bone marrow or spleen to become antibody factories. An antigen presenting cell, usually a macrophage, B cell, or dendritic cell, presents a foreign antigen bound to major histocompatibility complex 2 proteins on its surface to a T helper cell with T cell receptors on its surface. T helpers are incapable of phagocytosis, direct killing of infected cells or tumor cells, or secreting antibodies, yet they are so important to our adaptive immune system. Here we see a T helper cell with its T cell receptors activating a B cell to transform to a plasma B cell to make and secrete needed specific antibodies against the specific foreign antigen. Here, a helper T cell helps the B cell differentiate into either a memory B cell or plasma B cell. Our amazing antibody idiotypic diversity is obtained through the magic of recombination of the B cells in the germline theory, and more to be followed later in other videos of this fascinating topic. More than 2 dozen questions in the ACP Board of Certification Study Guide cover immune system physiology. Here are some similar questions on the various topics. Thank you for your time, attention, and support, and we hope to see you in the next video.